Hi, my name is Kamal and I'm a research technician here at UHN. Today I'm going to show you what my day looks like working as a research technician. I start my day at 9 a.m. and I begin my day by checking my emails and taking the time to make an action plan for the day. This is when I take the time to do any calculations I may need for my experiments on the given day. Today, I was going to help a student run a Western blot, so I went to the lab, put on my lab coat and went over the protocol from the SOP binder. One of my responsibilities is also to maintain this binder with up-to-date protocols and safety data sheets. Then we prepared our protein samples for the Western blot and moved on to loading the gel for the Western blot. Since the gel was going to run for about 90 minutes, I had time to prepare my mouse tissue samples for genotyping. The first step of performing genotyping is performing PCR to amplify the gene of interest, which is what you can see here. I then grabbed lunch before attending to my Western blot. By the time I was back, the gel was ready. Woohoo! I prepared the transfer sandwich cassette to then transfer protein from the gel to a membrane.
I then spent some time doing some data analysis. In my position, I'm also responsible for ordering reagents, chemicals, and plastics that are required in the lab. Well, this is the end of my day. But no 2 days are alike, so some days you might find me doing confocal microscopy, cell culture, ultracentrifugation, size exclusion chromatography, and going for lab outings.